I'm Jerry from Nigeria. I'm from Kaduna State. I had my BSc and MSc in ABU, biochemistry and nutrition, respectively. I'm currently on my PhD program at the University of Nebraska Lincoln under the Food Allergy Research and Resource Program, which is a food allergen program that is situated in the US. This lab, it's like the first laboratory that started food allergen research in the US. Okay, and I'm also a member of these below associations. They are very good associations. Obviously, I know you know about the Nigerian Institute for Food Science and Technology. And I and Immaculate were schoolmates in ABU. We did our masters together. So I really know her and I really appreciate the work she's doing in the food science space. So it's interesting to always be by her side when it comes to any food training or any program that will just make us to gain more knowledge on food and food science in general. So for today, this is the outline of our discussion. We're going to talk a bit about food allergy and allergens, then talk more about regulatory requirements, risk assessment and management strategies. Then we'll discuss on some analytical methods for testing food allergens. Then we have some brief introduction on proteomics, which is an area of research that is really kind of making headways in terms of allergen testing. And we're going to talk a bit about novel food proteins. These are recent information that are coming out regarding proteins and protein quality in different foods that have been consumed all over the world. Then we'll talk a bit about some emerging trends from food allergy testing and food allergens testing as well. So we can go on. All right. So food allergy. What do we really understand by food allergy? Food allergy are mostly immunological responses that are particularly triggered when we consume specific food proteins. And these proteins that are found in this food, they are like the primers for causing these allergic reactions that come up during some kind of in situations where you take a food allergen. The allergens that are mostly involved are proteins, and these proteins tend to trigger some kind of cells that release a kind of chemicals that triggers some kind of reactions. So we are going to see them, and these food allergies are mostly divided into three major types. But the one we concentrate more on are the IgE-mediated food allergy. These are what we call the immunoglobulin E-mediated allergy. We also have the non-IgE-mediated allergy, which involves immunoglobulin G, M, and A. And we have also a mixed kind of allergy reaction that might come up from IgE and cell-mediated reactions. So all these combines immunological responses and non-immune system kind of reactions. So for the IgE-mediated reactions, these are very fast kind of reactions that occur within mostly less than 2 hours after consumption of a particular food protein that is responsible for causing an allergenic reaction in susceptible individuals. And we can see that we have some kind of risks that are being attached to this kind of allergy. And first, we can see oral allergy syndromes. These mostly are kind of reactions that occur in the mouth, throat, tongue, and it can be swelling, inflammation, and itching that can come up. You also have reactions that come up from the skin rashes and hives, and asthma as well. And some of the severe and fatal kind of reactions are the anaphylaxis reactions, which mostly involve shortness of breath and shock. Those kind of reactions can lead to death. But for the non-IgE mediated, are not really kind of immune system kind of reactions, but one of the most particular one we can see here, or it's more popular, well known, is the celiac disease. The celiac disease is a kind of disease that is caused by ingestion of gluten, which is mostly found in wheat. And what happens is that when you consume gluten, the gluten triggers some kind of reaction. Okay. So the gluten triggers some kind of reactions in the small intestine. In the small intestine, there's what we call the mealy, they call them mealy. They are responsible for absorbing some of these nutrients. So what the gluten does is, for those that have the celiac disease, they don't have the mealy. So once you consume this gluten, it tends to not be able to be absorbed in the body, and this leads to a lot of variable kind of reactions within their system that might also lead to anaphylaxis or death and many other kind of allergic responses. So basically, these are allergies. What we basically know about allergies. So currently, there's no cure for allergy. So total avoidance of the food is what people do. So if you have an allergic reaction, or you know you're allergic to a particular food, the best thing is to avoid that food entirely. So that's why it's been encouraged that most people kind of look through food labels, nutritional information, to be sure that the food they are allergic to are not being added in the food product they are trying to consume. So how does the allergy work? What happens? So the first thing, there are two particular steps necessary for an allergic reaction to occur. The first is the initial phase of sensitization to a specific antigen, which is mostly our allergen. Then the second part is the elicitation of an allergic reaction after the second exposure to the same allergen. So in this sense, the first thing, if you consume an allergen that you kind of react to, the first outcome, you don't see any kind of symptoms, there's no reaction. But what happens is that the body gets sensitized to this allergen in particular, and this sensitization occurs by where the allergen attaches itself to the dendritic cells, and this attachment leads to the body creating a specific antibody. Normally an antigen and an antibody, when you have an antigen that is like a foreign introduction to the body, an antibody is being created to fight it off. So if the allergen comes in, the first thing is the immune system tends to produce a specific allergen antibody, and this specific allergen antibody are being attached to the mast cells. These mast cells are found all over our body, from the blood to our cells and tissues and organs. And once they get attached, that means the body has already recognized that an allergen is already in place, and it has generated an antibody that is supposed to fight off these allergens, right? So this is the first stage of sensitization. So what happens is that you don't get reaction during the sensitization stage. So until you consume this product the second time. So after a second exposure, the allergen now attaches to the antibody, which has already been created during the sensitization stage. And the mast cells that is found in our body have some kind of chemicals that are placed in them. These chemicals are like the histamines, the leukotrienes, cytokines, and the rest. So once the allergen comes in contact into our body for the second time and attaches to the antibody that was already created specific for that allergen, what happens is that the mast cells gets degranulated, thereby releasing these chemicals. So once these chemicals are being released, histamines especially, and cytokines. They lead to some of these reactions we see, the rashes, itching, and all sorts, inflammation of some of our organs, the lips, the mouth, the tongue, the neck, and all sorts. So this is what happens in an allergic reaction. I have a short video attached here, but I don't know if time will permit to play, so I can skip that. Um, so I hope we understand a little bit about the mechanism of food allergen reaction. I will share up the video. You can read more, watch it and learn more about the mechanism of allergen reaction. So for the prevalence of allergen, this is an overall prevalence of allergen in the world. And so this will help us understand how critical food allergy it is to the world. So we can see that the overall global prevalence for allergen, what you see on the red dots are mostly the prevalence for children, the blue dots are mostly the prevalence for adults, and the green dots are the prevalence for all other age groups. So we can see for children, we have about 2.4%, and the total, the world population is almost 8.2 billion people in the world currently, about approximately. So if you make your calculations, you can see that we can have approximately about 100, maybe 190 million children all over the world that are allergic to one type of food or the other. And that's also a high amount for adults. I know there's like a partial distribution of food allergy all over the world. Different regions have specific foods that they can react to. And most of some other regions, especially in Africa, we have a lot of unreported cases of food allergens. So we might not really see a prevalence data here. You can see Africa in general is just green. There's no much reported data on food allergies, so we can't really determine the prevalence. But I made a note about the US. The US and the US government are so keen on food allergy. And we can see in the US, the adults have about a 10.8% of the population of the United States are allergic to one food or the other. The United States have over 300 million people. 10% is about 30 million people. That's quite a huge number. There are some other countries that have as well higher population allergies, like you have Saudi Arabia. You also have Bahrain for children. It's quite high. So this is kind of a distribution. So we need to do more in Africa, especially in Nigeria. We have a lot of unreported cases, and we have people experiencing some kind of reactions that they don't even know is an allergic reaction. They feel it's just something that just came up, or maybe they just fell sick. And we always attach some kind of sickness or some kind of symptoms to some popular sickness like malaria, typhoid, and the rest. Some of these can be as a result of food allergens. So I think the regulatory authorities and the government will have to do more in terms of food allergy, because this is becoming an issue, especially for children that are being recently. We have high proportion of children coming up with huge cases of food allergy, something that the government need to work on like really well. So I remember during the pre-test, I asked a question about food allergy and food intolerance. I did the same. Yeah, so they are not. Food intolerance mostly caused by various factors. It can be deficient of enzymes. These, like, for instance, the popular lactose intolerance, we know it's when someone is deficient of the enzyme lactase and the body cannot really digest or break down lactose, which we know is a disaccharide, down to your glucose or galactose. And with that, it results to having some kind of discomfort in the stomach, generating gases, having bloated stomachs, and all such. So this is food intolerance. It has nothing to do with immunological reaction. So that's one of the major differences. Food allergy is an immunological response to consumption of a particular food protein. In particular, food protein. Why food intolerance can just be you not having an enzyme to digest a particular food. Must not necessarily be protein. As we know, lactose is a carbohydrate, so that's one of the difference. And sensitivity. So food sensitivity is similarly linked to food intolerance. Some people just call it the same. Yeah, but this food sensitivity can occur when you consume even small amounts. You can consume small amounts of food, and it might not really cause any sensitivity. You can consume a high amount that will lead to you having sensitivity to that food. So they are almost linked to food intolerance. So food allergen testing, how do we test for food allergy? So we need to, I'm going to explain through the course, the difference between food allergy and food allergens. So food allergens are particularly what causes the food allergy. So for food allergy, these are clinical testing that is being done. How do we determine that someone is allergic to a particular food? So there are various methods we use for this kind of testing. The first and most popular one is the oral food challenges, which mostly involves eating increasing amount of a suspected allergen under supervision of a maybe health provider or a medical practitioner. These are administered. Sorry. Okay, Immaculate. All right. So okay, we can continue. So these food are administered at time intervals. Mostly they use a 15-minute dosage interval. So they administer a small amount of the food, like a small concentration, then observe within a space of 15 minutes to see if there's any form of allergic reaction. And if there's not, they increase the concentration and administer again, then observe for another 15 minutes. If there is, they keep doing that till they see an allergic reaction, or if there is no any kind of reaction. So if you are doing the 15 minutes interval testing, you will know whether you are allergic to a particular food, and that helps to determine the kind of threshold dose that is required to cause an allergic reaction in that picture. Okay, and if you don't react to that when they get to the highest concentration that has been placed for that food, then you are not allergic to that food. So this is a good, very, very good way of testing food allergy. Then the second one is the skin prick test. The skin prick test is mostly called the puncture or scratch test, and that basically involves picking the skin with a small amount of the allergen and observing if there's any form of allergic reaction in the skin. So you can see, like this is like a kind of drawing that was done on the skin. So different concentrations of that suspected are being placed here on each section of the skin, and they tend to observe which part gets to react and what dose gets to react. So if there's no reaction to that food, you are not allergic to that food. If there's a reaction and they understand that you're allergic to the reaction, and they also know the dose concentration that might likely cause an allergic reaction when you consume. Okay, so these are very good tests used for food allergen testing. So further down our lecture, we are going to talk about how methods are used to determine these allergens. Okay. All right, so you remember I talked about food allergy, and I said there's also what we call the food allergens. So basically, food allergens are generally proteins, except in mild cases like sulfite, which has been identified by several regulatory bodies to cause allergic kind of reactions. Although they are not proteins, but they are assumed to cause some kind of allergic reactions, being mild or severe, like anaphylaxis. So most regulations tend to put sulfite as part of food allergens, but they are not proteins. But other than that, most food allergens or all food allergens are proteins. So these proteins tends to react with the antibodies that have been created. So if you remember the mechanism I discussed earlier, once the allergen comes in contact, you consume the food, the proteins in that food are what are responsible for the kind of immunological responses that are being induced. Okay. And in the US, sorry, I'm using the US. I'll still talk about international regulations for food allergy and also what we have in Nigeria. So in the US, we have 9 major food allergens that have been classified, and these cycled on red are the food allergens that are being regulated currently in the US. And mostly these allergens weigh between 10 to 70 kilodaltons. So they're quite large. Generally, we have proteins that are large biomolecules compared to other biomolecules like carbohydrates and the rest. Yeah, so they are quite large. And we have some emerging issues in terms of novel food proteins, which is part of my slides. Yeah, so here there's a question. So you can be thinking about that. So novel food proteins, are they threats? Are they threats to humans currently? Yeah, these are new food protein sources that people tend to consume. So we're going to discuss that further down. All right. So what regulatory requirements do we have for food allergens? All right. So currently in the US, there's the food that we call them the FACPA Food Allergen Labeling Consumer Protection Act, which was enacted in 2014 that listed 8 major food allergens apart from sesame. Yeah. So this act mandated that it requires that any intentional addition of any major food allergens must be clearly labeled on the food labels. So if you have any food allergen that is used in any of your food product manufacturing, you have to declare that food allergen on your food product. And this declaration can be done or must be done by listing these allergens by their common names. So for instance, you have an allergy, let's say egg, you are not going to list the egg, or if it's a fish, you're not going to list the fish with a botanical name. It has to be the common name fish so that it can be readable and clear for every consumer. Okay, so you must list that either as part of the ingredient. So in your ingredient list, you list this major allergen, or you have a separate what we call a contain statement. And this contain statement is, okay, you might just say this food product contains egg. This food product contains milk. So it's clear and stated. So these are intentional addition of food allergens in your food products. So they are part of your ingredients. They are what you use to make your product. So it's always advisable to that anyone that is allergic to such ingredient, we have to skip that food. Okay. So in addition, there's what we call the precautionary labeling. So the first one is the mandatory labeling, which is a must, is a requirement, but there's no for labeling or intentional addition of food. Okay, so what happens is that most manufacturers are being advised to put what we call advisory statements when it comes to cross contact. Cross contact is when you have a food allergen that comes in contact to another food that does not contain an allergen. But this contact is unintentional. It's not intended to be in that product. It might have come from, let's say, using the same warehouse or for storage, or using the same equipment for manufacturing, you understand, or not having a good cleaning validation protocol to clean off a particular batch of production before proceeding to the next. And there are many other factors that might be responsible for cross contact. It might also be the staff might come up with food from outside, and wow, it goes into your production line. And so there are many factors that leads to this unintentional addition of a food allergen. So they require, you are not mandated to do that, but you are required to state it in your product label that this food may contain. Because if I know I'm producing a food product that uses milk, and I know I'm producing another food product that uses cashew, which is a tree nut. And I know there's a probability that I use the same line or I use this similar storage facility to keep or store this food. So there's a chance that this food might mix or get in cross contact with each other. So once you know, even though you've done unnecessary step to avoid that cross contact, but since you know there's a probability, it's required that you put an advisory statement that this food may contain. Okay, and or this food is produced in a facility that uses this and this allergen. Okay, so and in the US, there's what we call the Food Safety Modernization Act, which mostly requires manufacturing facilities to identify and evaluate known hazards. These hazards are the allergens, so they are required to do that. So you have to create like a hazard plan to help curtail any unavoidable cross contact that might come into your production process. Okay. So what happens in the international space? So we can see here, this is just clipped table about some of the food that have been regulated by different countries. The first one is the US, which I mentioned. There's now 9 major allergens. We have the crustacean shellfish, egg, fish, milk, peanut, soy, tree nuts. So the tree nuts, encompasses of a lot of nuts. It can be over 100 type of nuts, but currently the FDA requires or listed about 23 nuts that are being regulated in the US. So these 3 nuts are my current research area. That's what I'm doing, working on my PhD, trying to develop mass spectrometric methods that will be able to accurately quantify these 3 nuts, especially when we have a multiplex function, as you have different nuts in a particular sample. We need a method that will be able to detect all of them and quantify them accurately. So that's currently what I'm working on. They also have sesame, wheat, right? So Canada regulates about 14 different, 14 different allergens. Yeah, so in addition, they also regulate mustard and shellfish. So the US do not regulate mustard. These are kind of regulatory fight that has been ongoing because there are some people that are coming up with new mustard allergen reaction. One of them, I experienced a mustard reaction when I consumed that, and so I tried to do like an oral challenge, oral food challenge to be sure, and it was confirmed I'm allergic to mustard, which is strange because I'm not allergic to anything. So it's just crazy, but yeah. So these are not regulated in the US, but in Canada they are. The European Union regulates more. They also have lupin and celery. So these are additional food allergens that are regulated in the EU. While for Nigeria, we regulate quite a number, but most of these numbers are also drafted from the EU guidelines. So in what's addition to what we regulate that is not in the US is the celery, lupin, and also gluten. Okay, so these are some of the allergens that have been regulated in the international space. So allergen regulation in Nigeria. So what regulations do we have in Nigeria? What regulations do NAFDAC have? Yeah, so currently our requirements are similar to that of the US and also the European Union, but there are quite some few exceptions or differences that I found out between the NAFDAC and FDA allergen labeling requirements. Like for instance, here the NAFDAC says there are some exceptions where the nature of food indicates it contains an allergen. For instance, a dairy product such as cheese, butter, or yogurt are clearly milk products, and milk will not need to be declared separately as an allergen. So what they are saying here is that I have I produce a cheese product or a yogurt product that I know I used milk, which is a known allergen to produce, right? So since we know that cheese is majorly milk, there's no need to label that on your product. There's no need to declare it as a separate allergen and say, okay, milk is contains allergen, or this cheese contains milk, which is an allergen. So this is the differences I saw from the FDA. The FDA requires you to label, even though you have a cheese or a yogurt, you need to label and make sure that the word milk appears in that label. And you can do that by putting, okay, if as part of the ingredients, maybe you're using a certain type of maybe cheese product and say cheese, you can put it in parentheses and say milk, or you have a separate contain statement that says this product contains milk. Okay, so these are some of the differences I observed within their regulations. I can send a link of the guidelines for food allergy for NAFDAC and that for the FDA, which I'll do that to Immaculate so she can share it to you. Then, so these are the listed allergens that are within the NAFDAC regulation. So we have 12 of them. And we have some of the 9 major ones. Then in addition are the mustard, celery, and lupins. Okay, reference doses for food allergy. So what do we understand by reference doses? These are most regulatory authorities and food industries usually base what we call their threshold level based on what we call the ED01 and ED05. The ED is what we call eliciting dose. So that's a dose that can cause an allergic reaction, that can elicit an allergic reaction or kickstart an allergic reaction. So the ED01 basically means the dose that is predicted to provoke an allergic reaction in 1% of the allergenic population, while the O5 is to provoke reactions in 5% of the allergenic population. So this chart was really developed by the WHO and the FAO expert committee on the risk assessment of food allergens. What they did was to determine, based on various kind of data that was available, to determine the eliciting dose at 01 and at 05. Basically, for a while, for some years now, they've been using what we call the eliciting dose of eliciting dose 01, which has been in place. So this is a dose that provokes reaction in 1%. So that's what has been invoked for over a while. But the expert committee sat recently in 2023 to actually discuss, okay, at revise like the ED dose, and discover these revision are based on a lot of kind of calculations from clinical data studies and also mathematical calculations. And they did that and observed that there are some kind of increment. So they need to revise and make sure that the food companies are ahead to now an ED dose that can provoke up to at least 5% of the population. So these are the dose level that is being suggested. It's still on recommendation though. So most food companies have not really shifted from the ED01 to the ED05 yet, but these are recommendations from the WHO expert committee that the food companies should shift their dosage level ED01 to 05, at least that can affect up to 5% of the allergenic population. So these are some examples of allergens and their doses, and also their reference dose, which is mostly the milligram of total protein from an allergenic source. So we can see here cashew, it's at that's the ED01 is 0.05. And there's a similarity note I did here. For instance, I just wanted to pinpoint this pistachio is a kind of tree nut as well, that is being regulated. So pistachio and cashews are botanically related, like they come from the same family. So they have a lot of homologous similarities in their protein sequences. So it's possible now they are finding new trends of what we call co-allergy, where someone that is allergic to cashew, but because of the close botanical relationship with another tree nut, that other kind of tree nut can also trigger the allergenic reaction, because these allergenic reactions are triggered by the proteins, and in the proteins there are specific areas of the protein sequence that really triggers this kind of reaction. So what happens is that if cashew and pistachio are kind of close botanically, that means it's possible they share some sequences that are similar. So what if the sequence that is responsible for the allergenic reaction in cashew is also found in that pistachio because of their close similarity? So there's no enough data that was available, clinical study data that were available to really pinpoint an ED dose for pistachio. So they had to use that for cashew, which because of what their close relationship and their possible cross reactivity that might occur. So this is basically on the reference dose and the threshold level that are available for the food allergens currently controlled. Okay, so we'll talk about risk assessment and some management strategies. So here I just really placed a guiding strategy just to guide us through some of the steps we're going to, some of the good manufacturing practice steps in terms of allergen control and management that we're going to see in the subsequent slides. The first, you can see the regulatory specified allergenic risks. So here, basically, is just to be able to identify what does the risks that might occur from an allergenic food. And also, we need to do the hazard identification of the allergenic ingredients. So pinpoint what particular allergen that is available in your manufacturing setup. So since we know the allergens have been regulated, so for every ingredient you're going to use, you need to understand, okay, this one is an allergen, this other one is not an allergen, this is an allergen. So you need to have a documented procedure that states this allergen. So at every stage of your production, from the purchase of the product down to storage, down to the processing, and down to packaging, you need to understand what might go wrong from the allergenic ingredients that are available in your production. Then you also identify possible cross-contact. So you look at the equipment line, you look at your storage facilities, you look at use of water or air dispersal that might come up within your factory. So all these factors must be assessed to be sure that, okay, we need to curtail or close down possible way that allergen can get in contact to a particular food that is not supposed to be there. So these are all intentional addition. Then the hazard control and monitoring and verification, and also implementation and training. So I'm going to discuss a bit about them in the following slides. So the first part is the product formulation. So what do we look at in product formulation? Supply management. So these may look trivial, but they are very key to managing an allergenic risk within your production processes. Because, okay, you have to review the product information and assess the risk. This is one of the basic things. Okay, I'm getting my raw materials from somewhere. I need to get these raw materials. You have documents. You are purchasing them from somewhere or importing them. So you need to understand, okay, these raw materials, are they at possible risks of any allergenic reaction? Are they allergen risks? So you need to identify these risks. Then create and ensure you create a plan to be able to cut or handle these risks. So from the supply management, that's where you start. From supplying, getting your products, your raw materials, your ingredients, that's where you start. You need to start documenting and assessing. So good documentation practice is a key thing in terms of control and management of food allergens. Then how do we handle raw materials. Basically, I told you that cross contact is one of the major key issue in terms of allergen risk assessment, because the normal intentional addition is well known. I add an egg to a food product. I know egg is an allergen. I know it's part of my ingredient. I'm going to definitely declare it on my food label. That's not a problem. And once you do that, someone allergic to that will read and know that he's not supposed to consume that. But the ones that come in contact unintentionally that you don't know as a manufacturer, that's what the key focus of risk assessment is. So now, how do you handle your raw materials? Once I get my raw materials, where do I keep them? If you look at your factory setup and some of the requirements by NAFDAC to set up a food production or plant or facility, there are basic outlets or rooms, that you need to have for your production. And I'm sure one of them is the raw material and packaging material room, where you keep your raw materials before you can now take them into the production material room for production. So these raw materials, if I produce variety of products, do I store them in one place? I produce a product that is milk containing, and also produce a product that is peanut containing, which is also an allergen. Do I store them in one place? So if I store them, how am I sure that there will be no cross contact that might come? Because any minute transfer of any of this, maybe the milk to the peanut, it gets into the food. And the doses, the threshold level for eliciting allergenic reactions are very, very small. Very, very. We are talking towards 20 to 25 ppm, which is very, very minute. So a small shift might trigger an allergic reaction in someone that is susceptible to that food product or that food ingredient. So we need to have a good, proper material handling procedures where you separate your allergens, keep them at far distance. You don't have space, you can create cupboards or wardrobes for them and just stack them in properly labeled. And once you're doing cleaning of your maybe raw material room, how do you clean? Do you just clean and make sure that the air, or if you're using a blower, it blows around and take the allergen from one place to another? So all these things are kind of the things you look at in your product formulation. So they are very key in curtailing allergen cross contact. Then manufacturing processes. Firstly, we look at the design. Okay, the factory design equipments are very critical. So the equipment design for is cleaning, because your equipment design is for the production of your food. So every food type have different kind of equipments that are being used. But cleaning is one of your quality control procedures that have been done. And I'm sure most of those food manufacturers do have cleaning quality control, which they should that they have in the facility for every cleaning that is done, maybe after every production batch or the rest. So how do you do the cleaning? They are very, very important. And sometimes you tend to clean with water, hope the water does not disperse to another place. You also clean maybe with dust, maybe cleaning dust. So some use kind of different air blowers and the rest that might pick up a particle of an allergen from one side to another ingredient. So all these are considerations that are being done. So we can see that it's advisable to use good ventilation systems like the HVAC systems, also even things like the dust extractor systems that help to pick up these dust and the rest, not spreading them around within your storage or factory facility. So, or the processing line as such. So cleaning is very important. There are very validated protocols for cleaning in terms of food allergen control. Then the production scheduling. So how do you do? Mostly production scheduling helps manufacturer to determine when to produce a product, at least to maximize efficiency and the rest. So how do you schedule your production? So that scheduling is very important. And one issue they have in terms of allergy management around food facilities is that most food facilities is a business, obviously. So they want to be highly efficient, so have high turnovers. So they tend to do like, okay, we produce now this batch, we don't give a rest, we produce this other batch. So this scheduling is done because most after every batch you're supposed to do a kind of cleaning, or especially if you are changing you're using a similar equipment for multi products, and you're supposed are using multi products, multi ingredients. So you're supposed after a batch for a particular product, you're supposed to have a cleaning procedure that is done properly before you go into another. So some facilities tend to just override that and just do a very shady kind of cleaning. And if you have produced an allergen, a food that contains an allergen in your first batch, and you don't do a proper cleaning, and you go into another one immediately because you need high turnover, that might be another way for cross contact. It's a huge way for cross contact that might come into your product. So these strategies are very, very important to emulate during manufacturing. Then the cleaning validation protocol, which is one of the key things in terms of avoiding allergen cross contact. How do we clean? The equipment we use to clean. If I use an equipment to clean a machine, how do I clean the equipment or the material after doing that? So it's very important as well, because if you don't clean what you used to clean, let me say in that word, it might carry over to another and to the next batch you're going to do. So these are key things. You have a cleaning validation protocol that is well documented, that can be followed step by step by your staff. So these are key issues. Here we should have to avoid air guns and high pressure water hose, because air guns are, you take them because you're trying to clean or blow off some particles in your equipment or within the production room. You tend to blow them into another equipment or into another area or into another product. So all these are very key. So you need extractors that can just pick them up. You understand? So that's why I mentioned the dust extractor system. They are very different type of extraction systems that are used in various equipments and various facilities. So all these things needs to be documented. So how do you clean? Do you dismantle your equipment according to the production schedule requirements? Like, do you have to, if there are equipments that you need to lose up before you can clean maybe the interior part, all these things needs to be done properly. Because if I'm producing something that has a miller that I mill, and before I pass the milled food or ingredient into another step of my production line, that miller, the inner part, how do we clean them? Do we just put water? Do we just put the extractor to take everything? We know they can't take. So do they, some facilities tend to lose equipment. So they have a cleaning schedule, they lose the equipment, clean it properly and recouple it back. So all these things are there. So we need to look at this when it comes to allergen management. This is one of the most difficult kind of control management that is seen in most manufacturing processes, as well as the microbiological management. So all these things needs to be done properly. And another thing is, to do this, you need to train your staff. So these trainings are very important in sites. You have to train them on awareness, how to identify and even know that this is an allergen. You understand? Train them on the prevalence, the severity and potency of allergens, and how do they handle materials. You have to train them on that, the equipment design, cleaning protocols, and all such. So you need to train them on that. And also one critical part is the personal hygiene that most facilities tend to avoid. And this personal hygiene is, I've done all the processes. I've followed all my documented protocol for cleaning, for storage, and all such. But because my staff are not really properly trained, so they might come with food, and we know there are places that you can't take food into production processes. So these things have to be continuously emphasized. Even in our clothing, you might eat something and to drop there, and you are there in the production line. You are not wearing a lab or a production apron or something close into your production line. So all these things needs to be checked. So staff need proper training when it comes to food allergen management, or any kind of production process or control management system that is placed in any food manufacturing system. Then the final one is the end product testing. So end product, we need to test. We are checking, okay, unavoidable cross contact. So we need to check to be sure that there is no cross contact that comes across when you produce a food product. Because if I have a batch of product that has been produced and I've already declared my allergens on that product, but I have other allergens that came into the product unavoidably, that's unintentionally. How do I know? You wouldn't know. It's not something that is written. It's not something you see on the product. So you have to test. So part of our testing for quality control, in terms of, sometimes I know most companies do send it to, some have testing facilities in their company, some send it to third-party testing facilities. So they test for nutritional composition, they test for microbiological assay and the rest. So allergens can also be part of what we test. Because if you're in an area where allergen is really, really controlled, so these are basic requirements that must be done. So what protocols are available, or what testing options do we have? And these, we have ELISA, LFD, PCR, and mass spectrometer. I'm going to talk a bit more about them, in the following slides that are coming up. So testing of our end products is very important to be sure that there is no any cross contact that may have come from an allergen that is not supposed to be in the product to the product we are trying to sell. So the methods, what methods do we use for testing? You remember the first time we talked about the food allergy, and that's the allergy itself. So we talked about some of the testing methods for allergen, like the oral food challenge and the skin prick test and the rest. So this other analytical testing is for our food allergens itself. So how do we test for them? So we have some methods that we use currently to test these allergens. So basically, we have the LC-MS method, the PCR. How we explain them properly. The PCR is the polymerase chain reaction, the ELISA, which is the enzyme-linked immunosorbent assay, and your LFD, which is the lateral flow devices. So this is just like a pyramid in terms of popularity. So from here, the one below, the LFD, which is the largest, it's like more in demand, more in use, followed by your ELISA, and then the PCR, then the LC-MS. So all these, there are various factors that affect the use and the rest. So we're going to see that. So for the ELISA, the LFD, which are lateral flow devices, generally they are regarded as common point of care diagnostics tools that are used. They are very, very easy to use. And they also use the immunological techniques or principles in their reaction. Sometimes people call them the paper-based technology that are mostly used for qualitative and quantitative detection of an analyte. So LFD has been used for various types of analytes, be it mycotoxins, also for allergens as well. So this technique is very, 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 very easy to use. There are some basic advantages for using an LFD. One, they are cheap, they are fast, They are user friendly, and you can also use them not in a basic or conventional laboratory, so they can be in field type of use. So because they are portable, you can take them to the field and just use them for on-site testing and the rest. So mostly the lateral flow devices uses the interaction between antibodies and antigen to detect the presence of these allergens. So basically, it's an antibody antigen reaction. That's why we call them immunoassays. So if you can see this cartoon above, you can see this is a basic principle of an LFD device. So it has four elements, which is the sample pad, the conjugate pad, the membrane pad, and the absorption pad. So basically, the sample pad is where you place the sample. You have a sample of a food product that you need to test. Maybe you're suspecting a milk cross contact. So you want to test that allergen. You have to basically, because it's a food product, you need to do an extraction. So you extract these proteins from the food. Yeah, so most of these LFD techniques have very simple extraction steps, very, very easy. Just sometimes a minute, 2 minutes, 3 minutes, or 5 minutes max, you're done with the extraction. So once you have that sample in the liquid form, you place it on the sample pad, then it flows down to the conjugate pad here. So the conjugate pad mostly contains antibodies. Yeah, so they call them conjugate antibodies, which bind to our allergens. So the conjugate antibodies bind to the allergen, forming a complex. So that's what we call the immunocomplex. So once it forms that complex, it now flows to the nitrocellulose membrane. So in the nitrocellulose membrane, it contains a binding reagent that binds to that complex that is formed by your sample and your conjugate antibody. So once it binds to that, so that interaction between our binding reagent in the nitrocellulose membrane and also the complex leads to a color change. And this color change can now then be read by the detector. So it can be mostly quantitative, and in recent developments, they are creating quantitative analysis for the LFD. So basically, so here, once it flows and there's a reaction, so if this reaction is positive, it just shows red and signifies, oh, this allergen is present. Okay, so the antibodies that are being raised here are raised against the suspected allergen. So if it's a milk, you understand, so they raise an antibody against the milk proteins. So that's where the reaction comes in and shows positive. So if there's no milk, the conjugate antibody will not bind to any of the proteins or peptides that is available. So there will be a negative reaction. So that's the basic principle for the LFD testing. So some of the issues we have with LFD, because the advantage, they are cheap, as we said, and fast, but the reproducibility is kind of very low because it doesn't require complex sample prep, clean up, and the rest. So there's a lot of matrix interference. Since it's a food product we are trying to test for, so there's a lot of interference that might come up from other components of the food. Maybe the carbohydrates, the lipids, and the rest. So those things might interfere with the reaction or the interaction between the conjugate and the sample. So these are some of the disadvantages for the LFD. Then the next one is the ELISA. So the ELISA, they are also immunological techniques like the LFD. So immunological involving antigens and antibodies. But in the case of ELISA, it's also linked to an enzyme. So as it's linked to an enzyme, it also requires a substrate to induce recognition, which can be used for the detection or for the quantification. So the ELISA techniques, we currently have four different types of techniques. or types of ELISA that are in place, which we have the direct, the indirect, the competitive, and the sandwich. These are different techniques that are being employed by different ELISA manufacturers. So most of the notable manufacturers are Neogen, we have Ad Biofarm, Morinaga, and the rest. So these, I'm sure you would have seen some kits for maybe mycotoxins or some other kind of analytes from some of these companies. So the ELISA technique is also very, very straightforward. It's interaction between your antigen and the antibody. So this is just an example of a sandwich ELISA. So what happens here? It's basically a wells of what we call the micro titer plate are being coated with what we call a capture antibody. So you can see these are the capture antibody. So once you add your sample, if there's an allergen or we call the antigen present, so once you apply and you add your sample, that antibody and that is being coated interacts with your allergen or your antigen. So that reaction here, they get, and there's an attachment which they react, they bind together. Then because the ELISA is an enzyme linked, the enzyme linked the enzyme is linked to what we call a secondary antibody. So this enzyme linked secondary antibody is added that binds to what the primary antibody that's already bonded to our antigen or allergen. So we can now do some washing to clear off those ones that did not bind. Then we now add a substrate. So because enzyme and substrate reaction, so because if you add a substrate to that enzyme that has been linked, then this can lead to what we call some color change. And those color change can be read by the ELISA reader, which are mostly colorimetric readers. So, and these are based on color intensity that are being read to determine the presence and also the level or concentration level of these allergens. So these two, the LFD and ELISA, are basic immunological techniques that are used. But currently in food allergen testing, in most food manufacturing systems, and also in most regulatory environments, the ELISA technique is the most used technique currently. It's the most used. It's easy and it's really, really accurate. Yeah, but it also has some kind of demerits because they have lower sample prep techniques. So there's a lot of metrics interference that might come up with an ELISA detection. So in allergen, we also have an issue when we use ELISA testing because, okay, you have a food product you want to test, milk, that method was raised for, let's say, milk, and that food now has been used, that milk has been used as part of an ingredient to produce a particular food, which has undergo maybe thermal processing. So there's heating involved. So if there's heating involved, what happens with heating? There might be change in epitopes in your protein. Heat might affect protein configuration and the rest. So now, because those ELISAs are being raised for particular peptide regions of your proteins there's your milk proteins. Now, because of those heating, if their reactions have occurred, let's say one of the most popular thermal reaction, which is the Maillard reaction. So the Maillard reaction is the reaction between your proteins and carbohydrates. So if that reaction occurs, you get what we call the Maillard reaction products. And these products now can be created at that specific epitopic sites which has been raised for that milk. You understand? So if those reaction occurs in that site, it means that once you use an ELISA, because the ELISA antibody are supposed to interact with those sites of the protein, but there's a modification that have now occurred because of heating or any other kind of processing. That means the ELISA will not be able to detect it because it cannot bind, it cannot interact. So these are some of the issues that ELISA have. There's a lot of matrix interference, a lot of issues when it comes to processing, especially thermal processing and the rest. So Analysts are now, all the scientific space are now looking for better techniques that can be used to analyze these allergens in an accurate and more precise manner. So that now leads us to things like the PCR, which is involved. PCR is quite good, very, very good. It solves one of the problem of ELISA, which is thermal processing, heat. It's heat resistant because it does not, the PCR testing does not involve protein. ELISA uses the protein. The PCR checks the DNA, because we know from the DNA, looking at the central dogma, the DNA get transcribes to your mRNA, the mRNA gets translated to your proteins and all such. So there's a link. So if you're able to understand the DNA of some of these proteins, where the proteins are coming from, you can be able to determine, okay, this is the DNA for a protein, milk protein that is coming, this is the DNA for a peanut protein. So PCR techniques have been used commonly, but there are also specific issues or limitations with the PCR method, because in the PCR method, since it does not test for proteins. So if you have products like oil, milk, egg whites, they cannot be tested with PCR because those products now do not really contain DNA. So it's very, very, very difficult to get them. These allergens, some of them will just be there. So they're just proteins. There's no DNA. They cannot really do anything. So it's when you get involved with biological samples, when you're using blood, you're using serum and the rest, that's when it's quite easy to use PCR. So PCR is kind of limited when it comes to allergen testing. But these techniques can also be there in some kind of allergy kind of testing, maybe once you're working on blood sera of patients, of allergenic patients and the rest. So you can use PCR testing for that. So this is basic. So now we have one of the trending method that is used for, or is invoked for, allergen testing. And this is the mass spectrometric method. So here we can see the LC-MS-MS, which means liquid chromatography tandem mass spec. So it's a kind of double mass spec. That's why we say MS-MS. Yeah, so this method usually work on ions, let's say ions in general. So when it comes to proteins, we call them protein ions or peptide ions. So what happens with the MS is basically it creates ions, separate ions, then detect the ions. So I'm going to walk you through the MS, since this is the trending method that have been developed for allergen testing, and I encourage, because mass spectrometry is very, very accurate. It can bypass some of the limitations from LFD and ELISAs, and also from PCR. When it comes to matrix interference, they are very robust kind of sample prep methods, use of a lot of highly important solvents for extractions, good solvents for salting and the rest. So these MS methods are very, very good. So you can really take out a lot of complexes or matrices in your food and just get a pure extract of your protein. So at that point, there will be no what we call matrix interference, because I've done a rigorous extraction just to get my pure proteins. So it's quite easy. And it's quite easy to use this MS for this kind of reaction. So I'll discuss more about the MS. So since we talked about the liquid chromatography mass spec, so I felt it's quite important to mention liquid chromatography. So the liquid chromatography is a separation technique, while the MS, which is a mass spectrometer, is a detector. It detects what you separate. So what we use the LC, which is the liquid chromatography, for is to separate our proteins or our peptides, right? Then once we separate them, using there are different phases kind of separation. You can use normal reverse phase liquid chromatography, normal liquid chromatography, or reverse phase liquid chromatography by using what we call our stationary, yeah, you can see them, and our mobile phase. Mostly the stationary phases are, they can be a lot of things. You have your columns, then your mobile phase are mostly solvents, organic solvents like methanol, acetonitrile, water, and the rest. So once you separate your proteins or your peptides, then you can now run them through the MS to detect them. So the MS now for proteomics, what do we use them for? So you can see the MS, basically, this is just a basic design of how the MS look internally. Yeah, so in your mass spectrometer, which is your, everything here is a detector. So just don't mind the detector here, it's just like a counter. Yeah, so what happens is you have your protein or your peptides. Those peptides are now ionized, changing them to ions, right? Because remember, I said the MS work with ions, so it detects based on mass to charge ratio of ions. So you ionize your peptides, then you analyze those peptides that have been analyzed, that have been ionized, sorry. Then after analyzing them, then the analyzing is to determine their mass to charge ratio. Then you cannot count them using the detector. So basically in allergen testing or in proteomics in general, so I'll talk a bit about proteomics next. So we use some kind of ionization techniques like your ESI, which is the electron spray ionization technique. And then your MALDI is a matrix assisted laser desorption ionization technique. These are very soft ionization techniques that are used. There are some kind of other ionization techniques that are used in other type of analytes. Maybe if you're working on, let's say, pesticide residues. Yeah, so you use hard ionization like the electron impact ionization, because these ionization types are coupled to gas chromatographies or chromatographs. So once they are coupled, you do your kind of very hard ionization using electrons, which basically just bombard your pesticide, break them into different ions, and then you now detect them through the MS. So, but in your LC, using any of these ionization is a very soft one. It doesn't break. The idea of doing that, we don't want to break the protein or peptide backbone. You know, the peptide backbone, if you look at, remember, I don't know, maybe those that have done basic biochemistry, remember the amino acid backbone, where you have your central carbons, your amino group, carboxylic group, your hydrogen, and your R group, which determines the type of amino acid. So if you use hard ionization, you might break up your protein and peptides and down to the amino acids and breaking the backbone, just creating different carboxylic and ammonium ions, which basically will not tell you any information when you're trying to analyze your data. So that's why they use soft ionization that will just ionize these peptides to give them that ion state that can be analyzed through the MS. So I remember we mentioned about proteomics. So proteomics is basically it's basically a large scale study of proteins. So any study of proteins is known as proteomics. So proteomics is used in a lot of fields, medical fields, when it comes to antibody research, cancer research, vaccine research, and the rest, food allergen research. So it's a very, very emerging field that is very useful for all this kind of research. And basically in proteomics, we have different approaches that are being used to proteomics. The first one is the bottom-up proteomics approach. We also have the top-down proteomics approach. Then there's one I didn't add here, which is what we call a native proteomics approach. I will just give you a basic difference. So what happens in the bottom-up proteomics? It's very simple. You have your proteoforms. Proteoforms are different isoforms of of proteins that might come up. So you know how, I don't know, maybe if I need to maybe refresh in terms of isoforms and the rest. You have a DNA, you have the DNA. Gets transcribed to the messenger RNA. The messenger RNA gets translated using the codons to your proteins. So in your DNA or in the gene, sometimes modifications happens at that level, or mutations. So if mutations happens, some of the nucleotides are being changed. So if you have a particular DNA and there's a mutation that changes one of the nucleotide, that means if it gets transcribed to the mRNA, there's a change in also the nucleotide, the comparable nucleotide for the mRNA. Then what encodes your protein will also change. So it might lead to a change maybe in one amino acid within the sequence. So you have a DNA that have gone to a protein, then there's a mutation, it goes to produce similar proteins, but with a deviation maybe in one amino acid or two amino acid sequences. So those two deviations are what we call isoforms. So these isoforms, when you combine them in terms of when they form a protein, let's say a native or quaternary protein, you have maybe three different types of proteins. You call them the proteoforms. So one DNA can encode different proteoforms. So now, let's say you have a protein. The protein gets digested. That's what the bottom up does. You get a protein, you digest your protein to peptides. So in your digestion, we use enzymes to do that. I'll explain that for you properly. So you break down your protein to peptides. So why we are breaking down to peptide is basically because we are using a liquid chromatograph to do the separation. It's sometimes highly difficult to separate these large, because I told you earlier on that proteins are very large biomolecules. So it's quite sometimes difficult to separate these large molecules in your columns. Although recently they are developing new column technologies that can really contain these large proteins, but it's really a difficult stage of data analysis using those kind of approaches. So, but basically, because they are large, we need to break them down to peptides. So once we break them down to peptides by digestion using enzymes, either trypsin or chymotrypsin or any kind of enzyme you might be willing to use, but the use of enzyme are very specific. You have to use an enzyme that is highly specific, that you can predict in silico the type of breakage that will come. If, for instance, now I have a particular protein, let's say of 200 amino acid sequence, and I'm using a trypsin enzyme. Why I'm going to use a trypsin enzyme is because trypsin enzymes are very specific to breaking bonds at lysine, after lysine and after arginine. So because they are specific, I know that it will not break at any other amino acid sequence except lysine or arginine. So since with that, I can really do an online prediction of the kind of breakdown that might happen. So why it's important is because if you come to data analysis, you've done an experiment in vivo or in vitro, let's say, and you are now trying to determine those peptides. You won't know. This is an experiment. So how do you do them? You have to match them to something that is known to be able to get or identify those peptides properly. So basically, we break down to peptides for easy separation. After separation, we now send them to, we now do the identification by matching them to online databases. So I will explain that properly. There's a slide for that. Then the other approach is the top-down proteomics, which people do. It depends on the type of kind of research. For the bottom-up, it's in terms of food allergen research, it's the most widely used. It's far better. It's easier because we are working on food matrices. You understand? So it's easier to break down these peptides, then we identify them back and infer the proteins they came from. Why for the top down, when you're working on smaller proteins like like let's say vaccines, yeah, like vaccines or intact proteins. So, and you want to understand maybe different, you're not just trying to check whether this is present, you're trying to identify maybe binding sites, you're trying to understand the protein surface and all such. So you don't need, if you digest such such proteins, you're going to lose the native state. So you will not be able to do some kind of identification like the binding sites and structural elucidation of those proteins. So that's why they use the top-down approach, which they run the protein without breaking to peptides directly to your MS and identify the protein forms. While in the native MS, basically the native MS, it's a new one that is coming up. Not a lot of people are in that area, but it's when they are trying to, it helps them analyze the proteins in their native state. That it has all, if it's an enzyme, if you're working on an enzyme, you want to understand the enzyme in the native state. You want to understand the structural information that might come up and any other complexity that might come up within the protein. So you can use what we call the native MS for that. So these are the three major approaches in proteomics. So basically for our bottom-up approach, which we use in food allergen research and testing, it's basically, I told you, we have to digest. So for these proteins are very diverse and they have different amino acid sequence, which means they have different masses. So now you can see one of the limitations I mentioned, that the MS, they don't work well on very large molecules. So that's why we need to break them down to, you can see here, you have a sequence of protein, you break them down to different peptide sequences. So once you break them down, you have the M in the ionized state. So with them in the ionized state, you have their masses, then you can be able to analyze them using the MS. So most of our digestion are done by enzymes. The most popular one is the trypsin enzyme, which is mostly gotten from porcine. So it's pig. Yeah, they extract them from pigs. And those are some of the high-grade sequencing trypsin enzymes that we use in the lab. So these are used because they are highly specific. As I told you earlier, trypsin cuts after lysine and arginine. S7 proline is the next residue, but basically lysine and arginine. So since we know their specificity, it's easier to predict their breakage using in silico or bioinformatic tools. So we can be able to do what we call the experimental matching. I run an experiment, use trypsin, break it, then run a computational experiment, use trypsin and break it. Then I can be able to match those two, the data from my lab experiment and the data from my online or bioinformatic experiment. I match those two together, and okay, this one matches to this. It has the same mass to charge ratio and ion state. I match, I identify, this is the peptide. Since in my lab experiment, I can't see them, but in my in silico experiment, I can see the peptides. I can see the protein breaking the peptides. I can see it and say, okay, yeah, these 7 sequence amino acid peptides are created, 25 sequence amino acid peptides are created. These are them. You understand? So if I match those two information, I should be able to predict the peptides that are available. And once I understand or identify all the peptides that are available, I should be able to use infer their proteins using bioinformatics. So there are various bioinformatic tools that we use to identify such proteins. So this is just the general flow of, you have your sample, your proteins, digest to peptides, run them through your MS, you get your parent ion. The first ionization that happens, you get your parent ions. So the peptides are ionized, you have an ion with the charged state. Then we now further fragment. That's why when I was mentioning LC-MS, I didn't mention LC-MS, because LC-MS is single MS type of equipment, while the LC-MS is a tandem type of equipment. It's just like having 2 MS in building one. So what it does is you have a first ion that is being ionized. So you don't read the ion, you further break down that parent ion, or we call them the precursor ions, down to fragment ions. So the essence of breaking them down is to improve like the specificity of our identification. So if we break them down proper to smaller ions, it's easier for us to be able to denote where they are coming from. So that's the essence of using a tandem MS. So it's very, very important in proteomics. So here you can see from masses to, that's what I've been trying to explain the other time. You have your protein. This other left part is the laboratory experiment. Then here is your computational or in silico experiment. So you have your protein here, you digest with your enzyme, you run it through your MS, you get your data. Then you go to use your computational tools, you digest. So there's what we call the peptide cutter. Is a tool online, it's free. So you can use that. So you digest, you get your theoretical data, then you now match the information from the two data. So if your MS1 data, which is your parent ion, and your MS2 data, which is your fragment ions, you match them with what you have from the experimental data. Then you'll be able to identify, since I'm in the experimental data, sorry, in the in silico data, I'm seeing my peptides, so I should be able to identify them and know which peptides were available in my experiment. So the idea is to identify every peptide that is available. And once you identify your peptide, like you see here, then you can now infer your protein and know which protein is coming from. So if you know the protein is coming from, you know which allergen source it's coming. So if I'm able to identify a peptide that is coming from, let's say, casein. So I know casein is a milk. So I should be able to know that, oh, there's a milk allergy present in that food. So that's how we do the allergen identification using LC-MS. So it's a very, very innovative tool. It's accurate, it's highly robust, and it's highly specific. Those are the advantages. The only disadvantage we see with the mass spectrometric method is they are very costly. They are very huge equipment, so it requires a lot of money to buy, a lot of money to maintain, and the maintenance is one of the major things. Like, I think it's very, very critical, because if you want to be very accurate, you need to maintain your equipment properly to ensure that it runs or work at the optimal level that has been required. So that's just the disadvantage. But other than that, it's a very, very wonderful method to be used, and that's what a lot of analysts are using in research. And we have in the US here, they have manufacturers that are having testing laboratories that have LC-MS for allergen testing. So it's quite refreshing to hear. So I said I was just going to just give a brief about novel. You remember one of the slides earlier on, I said you should think about novel food proteins. So why is it important? Why is novel food protein important? Because now we know the regular allergens that we have, right? They are all listed. By now, everybody should be able to mention the allergens that are available or controlled in Nigeria, let me say, not the US, yeah. But we know the major allergens that are available. So we have our conventional fish, egg, tree nuts, peanuts, milk, sesame, and all such, wheat, and the rest. So now, the new trend is that people, because of protein deficiency and malnutrition, people are now finding innovative, or not, let me not say innovative, people are finding alternative sources of food or food proteins, right? So they are now looking into other type of food that are not conventional food that we eat. And these other type of foods tend to have or tend to be very rich in proteins. And as long as they have proteins, that means there is a tendency that they might have allergenic residues in them, right? So these are huge concerns to those in the food allergen space, because now they're introducing new foods or new alternative novel foods that people don't really eat or don't know about. And you might be experiencing an allergic reaction and might not be able to pinpoint where it's coming from. So these are some of the ideas people are coming. So these are novel ideas that those in the allergen space are working towards novel food proteins to understand them better, understand the protein structures, understand, do a lot of clinical testing and all such. So here is just a chart just to see, like, okay, this part where my laser is, ASF, they are animal source food. That's the normal animal source food we have, fruit, which are your meat, your fish, your egg, and the rest. So you can see. Then this one is your novel food or future foods, we call them. Then the plant-based food. So you can see the shapes. So looking at this trend, you can see the plant-based food are mostly having high protein content, right? Why you see the novel food are also working. They're also following behind, let me say, while your animal-based food are still down here. So what it's telling us that these novel food proteins are having higher concentration of proteins as compared to our conventional animal proteins. So this is a table that gives a clear example. Here is a traditional protein source. Pork, you're having an average of 22.2% of protein. Your beef, 26. You can see them here. While looking at some of the novel food proteins that are now in vogue, the consumption of microalgae is quite huge. Quite huge now. It's becoming a new thing. It's becoming a really new thing in, I don't know for Nigeria, but I'm sure here in the US and some of the EU countries, and they are becoming a new thing. So you can see the concentration ranges, the protein concentration range from 30 to 70%, which is a huge amount. So imagine having this huge amount of proteins in a particular food. You can't just take out the probability of not having an allergenic site within those. So that's why a lot of studies are coming up regarding some of these things. So this is just for this. So you can see the protein percentages in novel food protein sources are higher compared to the traditional food proteins. So these are some of the sources, that covers the novel food sources. You can see microbes, bacteria, yeast, algae, fungi. There are a lot of products that are coming up with these proteins. Insects. Now we know that in Nigeria, I'm sure we eat a lot of insects as well. The grasshopper, what was this other one we call, and I can't even, is it the something termite, shingi? Yeah, so these things are, they are insects that we eat. You go to the Asian region, they eat a lot of insects. Yeah, although the insect consumption are kind of lower in most of this Western world. So it's there. So these are some alternative food protein sources with high protein content. Your plants, which are the normal ones we eat, and genetically modified foods are also there. Then you have cultivated meat. Cultivated meat is now a trend. And these are lab-grown meats that are produced by normally collecting cells from living animals. Then they proliferate the cells, then and make them into meat by using techniques like cellular agriculture. So those are there. So these things are made in made in the lab, and they are available. People eat them a lot. They taste like the normal meat because they are gotten from the cells of the normal meat. They taste like the normal meat. They smell like the normal meat. They have the proteins, similar proteins, even more improved protein concentrations and the rest. So, these are a new trend. Then you have the marine organisms like your microalgae as well, and the rest, seaweed and the rest. So these are good protein sources. And even there's what we call a novel food protein process. That's novel food process. These are byproducts that come during processing of our normal food. So these byproducts, like they use like the hawks and hull, normally we don't really consume them. So these things are known to have high amount of proteins. They've been tested to have. So people are now looking towards them as alternative source of proteins and food. So these are also notable additions to the novel food proteins. So um, it's just quite interesting to just look at these things. So what concerns do we really have? Yeah, the novel proteins can have a lot of concerns. We can say I listed some here, some anti-nutritional factors, pesticides, mycotoxins. These are huge concerns for novel food proteins. But for the scope of this study or this discussion we are having, we focus on the allergens. So the allergens are also a concern. Yeah, there's still a lot of work to be done regarding this, but they're concerned. So that's where we come with issues of co-allergy. Although in our conventional allergens that we have, like the tree nuts, we're already aware of some co-allergy. I mentioned earlier about cashew and pistachio. So you can see this is a phylogenetic tree that you can use. Also, if you want to design this tree, you can use there's this thing we call interactive tree of life. It's an app. It's not an app, it's a website that you can just go on Google and search. You can connect. It's easy to use. So you can use that to design similar things if you want to connect botanically related organisms, whether plants or animals or bacteria. You can use these to design something like this. So this you can see in this family, the Anacardiaceae family, which cashew and pistachio are there. So they are highly closely related. They are within the same family. So because of this close relationship, there are probabilities that they share similar amino acid sequences. They might have similar amino acid sequences. So if they have similar amino acid sequences, that means the allergenic epitopic sites that are on cashew might also be there on pistachio. So if, for instance, I'm allergic to cashew and I now start consuming pistachio, because there's a probability that pistachio might have similar homologous sequence with cashew, that means there's a probability that it might also elicit an allergic reaction in me because I'm allergic to cashew. So that's the idea of co-allergy. So people are now working towards, so for my own research, working towards these tree nuts. There are 6 tree nuts: cashew, pistachio, pecans, walnut, hazelnut, and almonds. So the idea is to be able to get a method, a single mass spectrometric method that will function in a multiplex manner to identify all these nuts in one single run. So issues of cross contact, if I'm producing a food that has cashew and peradventure there's a cross contact with almond that you don't know, but because I'm trying to test, I can just use that method if developed to test, and I'll be able to identify if any of these 3 nuts is within that food product. So that's my current research. That's by the way, but these are what we call the allergen cross contact. So if there's an co-allergy, there's co-sensitivity and there's co-reactivity that might occur. So these are potentials. So another one that we look towards novel food proteins that we've seen, and there are some articles on some research that have been done, is there's a potential cross-reactivity between crustaceans. You remember crustaceans, shellfish, are one of the major allergens all over. Yeah, so you have them, oysters and the rest. They are one of the major allergens. So they also have potential cross-reactivity with insect because of this botanical relationship. So we can see here, I just put in a slide of allergenicity in edible insects just to understand. So here we can see that insects are part of a phylum of animals called Arthropoda, found in this. Yeah, so this Arthropoda, you can see the phylum here. So you can see where the crustaceans are, and crustaceans are, we eat a lot of them, crab. So you can see here Hexapoda, where you have your insects, the Myriapoda, where you have millipedes and the rest. So these are some novel food choices for people. So a lot of people are eating a lot of insects and a lot of marials. So you can look at this, that because they come from the same phylum, that means there's a probability of cross-reactivity So people are looking towards that. And when you look at cross-reactivity, we look at the proteins. So there are some research that have been done to determine the proteins that are available. So we know the proteins that elicit allergenic reaction in crustaceans are the tropomyosin and the arginine kinase. These are the two major proteins that elicit allergenic reactions in crustaceans. Is a known fact. That's why crustaceans have been added as the major food allergens. So now, these tropomyosin and arginine kinase have also been found in some of these insects. Yeah, so the exact type of proteins have been found in some of these insects. So that potential of cross-reactive or cross co-allergy is there. So how do we curtail this when it comes to novel food proteins? So that's why currently a lot of research is coming up with regarding novel food proteins, just to ensure that our food is in a safe space. Because if peradventure you have some of these novel proteins come into a food product and they tend to cause co-allergy, then there's a problem. So that's why we need to look into this area of novel food proteins. All right, so I'm almost done. Um, so I said I was going to just talk about some emerging trends in research. So for this, I'm going to just talk about emerging trend in food allergen detection, then emerging trend in food allergen detection. So this is for the food allergy detection. Remember previously the conventional methods, we talked about the oral food challenge, the skin prick test, and also the oral food challenge and the skin prick test. So now these are some innovative techniques that are coming up recently in areas of the allergy testing. So you have the basophil activation test, which these tests kind of use the idea of flow cytometry to measure the expressions of degranulation. Remember when I talked about the mast cells in the allergen mechanism, I say there's a degranulation of your mast to release histamines and the rest. So that's the same thing that happens here. So they just use the flow cytometry to test those degranulation markers that come off from your basophils and from your mast cells, sorry. So once they measure those ones, they can now really determine, oh yeah, you are allergic to this product. So this is similar. So the basophil and the mast cell activation tests are similar. They just do it on basophils and mast cells to determine if someone is allergic to a product. Then you also have the bead-based epitope assay. This is a recent one that utilizes like two high informative sequential epitopic cells. So what they use is that they use like an highly sequential epitopic antibody, mostly their immunoglobulin E antibody, to predict the outcome of any oral food challenge. So it's like in conjunction with the oral food challenge, but it helps to better predict what might happen when you take in a particular allergen for this time, after this time, and this. So they are innovative techniques. So now we are seeing things like using AI and machine learning to check for allergens. Yeah, so these are coming up, something like the diabumine, that it has been used now for milk, egg, and peanut allergies to help determine if newborns and up to 3 years of old babies are allergic to either milk, egg, or peanut. So you can use some mathematical formulations and computer-based kind of program and computer-based kind of how will I put it? Yeah, like let's just say computer-based kind of processing here to just determine if newborns from 0 to 3 years can be allergic to any of these products. So they've created a diabumine for that. And also the clustered regularly interspersed short palindromic repeats, CRIPS, they call it CRISPR. There are technologies that have offered groundbreaking methods to edit gene allergen genes. So what they do is that they use these methods to edit a possible gene that might lead to allergy in a newborn. So these are techniques that are used in clinical space to help correct possible or future allergy of a child or a newborn. Or so these are new innovative techniques. So when it comes to the allergen itself, the testing, we also have innovations that are coming up. We know that the use of the tandem mass spec is the gold standard, but there are other things that will assist this tandem mass spec in creating or getting more accurate and more highly robust kind of results. So we have these the biosensing techniques, which the biosensing techniques are mostly lateral flow devices too. Yeah. So, but now you remember in our conventional testing we use the lateral flow device. So now to improve the accuracy, they are now infusing nanoparticles to creating biosensors that will help. So this is an example of the aptamer functionalized gold nanoparticles that are used as probes to test some of these allergens. Then we also have the PCR methods that are used now in a multiplex manner. So they can use those methods to test different allergens in one single run. Then we also have the high resolution mass spec, that they are super high resolution. And there's this idea of creating a portable mass spec, which is still a long way off, but is at a beginning stage. So a lot of companies are trying to work, mass spec producing companies are trying to work towards creating portable mass spec to make it easier for people. So those mass specs will be kind of a bit cheaper and available for facility or for manufacturing facilities to be able to procure and maintain. Then the idea of quantitative MS. So basically, most MS methods currently use just the detection. They are just used to detect the allergens, but because of their high accuracy and robustness, you can be very sure that you are getting the right results. So now the new trend is a quantitative MS, creating a quantification method by using what we call the stable isotopic labels. So we use that as a yardstick to create a method. So for my, sorry, I'm getting into my research. For my research, I'm working towards quantitative MS. So basically, if I digest my peptides and I know my peptides, and I've done a lot of screening, filtrations, and the rest to select what we call target peptides that can be used for my method development. For me to develop a quantitative method, I have to create a synthetic peptide of my target peptides that I've selected from my digest that I've selected from my digest. So once I know my target peptides, I send and maybe just we normally collaborate with some official to do that. So we create a synthetic peptide. We call them the heavy peptides. So what it is just to add an extra, have a heavy nitrogen. Nitrogen exists in two states. So you have the light 13 entry N12 and we do the N13. So we create the heavy nitrogen. So now of the same peptides that I've selected. So once we do that, we can now use the relative ratio between our light peptides that we've gotten and the synthetic peptide to do quantitation. So that's basically how the quantitative MS is going to run. But it's a new trend. So we don't have a lot of methods that have done quantitative MS. So, but we're coming up. So basically, these are some of the emerging trends that we have in allergen testing. So thank you for listening, and I'm open to questions, discussions, and
Good evening, and thanks so much for the wonderful lecture.
thank you.
Yeah, I just want to know, when it comes to allergic reaction, what really matters? Is it the presence of the causative factor, irrespective of the quantity amount present? I don't know if you understand.
Okay, you can rephrase. Just ask it again. I didn't get
Okay,
the first part.
okay.
Your
Like
voice is kind of a bit
if
low.
we have,
Okay.
okay. Can you hear me now?
Yeah, better.
Yes, we were talking about allergic reaction.
Yeah.
There are some certain kind of food, like for instance, now if you're talking about milk, you say
Okay.
we have lactose-free milk,
Mm,
and we also have some that's actually they are not lactose-free.
mm.
Now, are we looking at it now that if the of the hello,
Yeah,
so the
you
network is
skip.
breaking. Can
Yeah,
you hear me?
I'm hearing you. Thank you.
Okay. Now the quantity of the allergy present,
Yeah.
is that the one that counts or not even present at all? For
Yeah.
instance, now if the if the maybe we are talking about some of these causative allergens now and allergic reactions, if they are there, is that when the reaction will occur or the quantities that determines it? That's it.
Okay. Yeah. Thank you. So, in allergic reactions, like for instance, from the known allergens that we have, if you consume an allergen source, you consume everything contains the protein. So let's not forget the allergens are proteins, right? The
Yes,
allergens are proteins. So
yes, yes.
if you consume a milk, for instance, you are consuming the entirety of the proteins present in that milk. So now the allergic reactions, what causes the allergy is not the entire protein in the milk, but there are specific protein regions, or we call them epitopes or amino acid sequence within a particular protein, you understand, that will
Yes.
cause that or elicit that reaction. So sometimes the reaction is being elicited based on dose level. So concentration matters. So that's why when they are doing a prick test or the oral challenge, let me say, they give you from a lower concentration to a higher concentration. You remember I said that? So you might take for a particular allergen and depending on the person. So it's not like a unified value for everybody, but depending on the person, you might take a small concentration, let's say just hypothetically 0.5 ppm, and it might not cause or elicit a reaction. But that level to let's say 2 ppm might not elicit that reaction. So if those concentration dependent, the allergen is there, it can cause, but because it can also be influenced based on the concentration of that allergen, right? Then another person as well that is also allergic to that milk can take a 0.5 and the allergic reaction will be will be induced. So it's dose dependent and based on individuals because we have different physiology and the rest. So, but what they do with threshold levels is that they determine the barest minimum. You understand? The barest minimum concentration that might induce allergenic and that might induce allergenic reaction in an allergenic population. That's why I talked about the EDO1 and the EDO5, which are eliciting dose. Like the EDO1 affects about 1% of the allergenic population. So if an EDO1 affects, you set the dose at EDO1 that affects, let's say 1% of the population, can elicit, allergenic reaction at 1% of the population. That means 99% at that dose, when they consume that food, will be affected or will have allergic reactions. So it all depends. So, but what you have to know is that it is concentration dependent.
Okay. Okay. Thank you very much.
All right. Thank you.
Is that all, Femi?
Yes. The other question I would love to ask is
Okay.
because there are several instances like that. I actually work in an hospital,
Okay.
so we have several instances of patients coming in, and then they have this allergic reaction, and we find it very very difficult to manage them because of the allergic reactions. Because definitely there are situations they actually demand that they eat more protein.
Mm.
But we find it difficult to do that. So as a result of that, we have to run a survey, and we notice that this allergic reaction is common with people that are those rich and well-influenced people in the community. I really want to know, do you have an answer to why this thing is somehow common among this
To
set of population?
that's to those in affluence.
Yes,
Okay. Yeah.
because
There's no scientific justification for that, though. But the only thing I can think of, right, permit me to say, it's maybe access to a, because these proteins are that causes these reactions are rich, like they are kind of expensive, right? Normally you look at things like fish, crustaceans. A lot of people don't eat crustaceans because they're expensive. Yeah, you have, although fish, milk, and the rest are pretty common, and soy and the rest, which you look at them, tree nuts. A lot of people don't really eat tree nuts. So all these things might be access. So because they have maybe the influence to get more of these or different varieties that might come up, so the probability might be a bit higher for them. But that's just the response I might think. But there's no scientific justification for it being maybe tending towards a particular set of people in terms of affluence or in terms of maybe rank or status quo. So yeah, but basically maybe because of access to some of what maybe others might not be able to get. So okay, like for me, I gave an example with myself. I mean, I was in Nigeria. I've never eaten mustard. I don't know. I've eaten all the allergens. There's nothing. I've never eaten mustard. But I came here and ate mustard. The first time I ate it in a mustard cake. The cake they made, then I added mustard. I was in the office, then I started feeling some kind of reactions from my throat. So it was getting a bit bloated. Then I started having difficulty in breathing. So I had to even go out of the entire building just to get fresh air and everything. So I was feeling, okay, maybe I'm kind of allergic to mustard or something. So I just said, okay, let me try and see if we can do, since we have the facility here in our program. So we just went, I just did. And voila, I'm allergic to mustard. So you can see, I didn't have access to mustard in Nigeria, but I had it here now. And all of a sudden, I'm allergic. So that's the thing. So it all depends on the food source, the geographical location of the food source, the type, the access to the food source and everything. Yeah, but in terms of physiology, it's kind of an equal game for everyone.
And perhaps we can also look at it this way. In Nigeria, for instance, we have high level of food insecurity, so to say. So just like we would say, no be person wake up, go they think of allergens. At least I've heard that couple of times. Each time we try to sensitize people, you understand? So perhaps the people that have access to your hospital are the rich, so to say. The average, maybe the lower class, citizens, perhaps they don't have access to your hospital. That could be the fact, but I don't think it's a thing that has to do with any class. It affects all class. Okay. So we have Jasmine. Your question.
Okay, good evening, everybody. Thank you so much, Mr. Kuku.
Okay.
It reminds me of good old days of biomedical pathways.
Okay.
So, please, my question is, in fact, there's been a lot of things that have been puzzling me here in Nigeria.
Okay.
And it gives me cause for concern. First and foremost is that you actually find out that you are, what do you call it? You're exhibiting some symptoms, which can be physical or what do you call it? Physiologically, as in within your system or on the surface. On your skin and all that. And that wasn't the case before. And it gets to an extent where it becomes very uncomfortable. And some of them, you start having a really reaction, like maybe kind of a bit of itchy, uncomfortable sort of reaction. That something is not right. Something is out of place. And you go to your physician, as in to the doctors, and they take your history, and you're expecting that they should be able to run. In fact, the predominant test they will run is widal test, malaria tests,
Yeah.
And most times, yes, a typical thing test they will run here in Nigeria. My question is there no precise and by the time they run this test and find out that you don't have enough plus reactor or reaction to show that you have malaria, or there's no traces of widal test positivity,
Yeah.
And then they'll start asking you your lifestyle for the past
Mm.
how many days, what you've been eating, and what it becomes so difficult because you eat a lot of things which
Mm.
you're supposed to eat as a normal human beings and
Mm.
a Nigerian.
Mm.
Now they're asking you questions to be able to single out or filter out what could be the cause or thereabout.
Yeah.
And my question now is, have we not gotten to the extent, to that level of exposure or technology that you go to a hospital and they suspect some allergies and all that, they could actually use, there are tests or
Mm.
what do you call it? Yes, tests they could
Mm.
run,
Mm.
examinations that could be conducted, and they're able to rule out this and say, oh, this is likely the cause, instead of guessworking, because that's what is done. They just do guesswork.
Yeah.
And at the end of the day, they give you anti-allergy, antihistamine drops,
Drugs.
like loratadine,
Yeah.
to clear the allergy. Or,
The
I mean,
symptoms.
it's too risky. Is there
Yeah.
no definite procedure or test that can be carried out to say, oh, you are allergic to this or allergic to that? It's baffling. It's mind-boggling. You keep going and they keep pumping you with antibiotics and not finding out what you're allergic. And when you finish, you go back to that thing you're eating, and the same thing comes up, and you know it, and you go back, and they don't give you a definite, a finite answer. So, is there no tests that can be done in Nigeria to say, oh, for you are allergic to this as an individual?
Yeah,
That's
good
question number
yeah.
one. I still have
Okay.
another one after this.
Okay, it's fine. So thank you for this question. It's really this really important, because the truth is, it's the healthcare system if you look at it, because these tests are available all over, and some of these tests are quite cheap. And like I mentioned earlier about the testing, I mentioned about oral food challenge. Oral food challenge is there. What you need to do is, okay, you're taking an oral food challenge, and it all depends. Okay, let me just start from this point. If I feel it, remember from my introduction, I mentioned most of the allergic reactions are IgE related. That means immunoglobulin E induced. And they occur within mostly within, let's say, 1 minute, the highest time you can record it, maybe 2 hours. But most of the allergic reaction occur between 1 to 30 minutes. That's from the well-known study that has been done. So if someone consumes a food and within a short period of time feels some of these reactions, just like I felt when I took that mustard cake, like I suspected, but because I had the knowledge, so I suspected, okay, maybe it's an allergic reaction, so I need to do something. But if you have these kind of reactions, you understand, now that depends on the people. Like, we need a lot of information to parents, to individuals, to the population about allergy. A lot of information needs to be delivered, because now if you look at most of the onset of allergy starts from birth. A lot of children have the highest susceptibility of getting allergic reactions. Some develop at adulthood and the rest. So it all depends. But because of lack of information, we don't really know that some of those reactions are due to allergies, not due to another sickness, or not due to chance, or let me say village people or something. But at the end, once we understand those things, then it helps the health facility to determine, okay, I have a reaction within this. You can be able to discuss with your health providers and say, okay, this reaction happened because within a period of, maybe you ate something it happened because I ate something within this. So would there be a possible cause that this reaction might have come from this food that I've consumed within this period? So you can able to pinpoint. So if you are able to pinpoint, and these facilities are available in the hospitals, which I believe there are some hospitals in Nigeria that do have those things. They have those assets, they have those testing facilities. So they can now say, okay, these are the ingredients that were used. Which are the known allergens in these ingredients? They pick them and say, okay, we can now do an oral food challenge for these things you consumed. Then they can now test, and confirm, right? So one, from your question, knowledge about allergy is very important, not just to the experts or to the health facility or providers, but also to the people, to the masses, it's very, very important, because if we understand this, then we'll be able to tackle it easily. So imagine you've gone and after 2 days, they're asking you to give a recap of the food you ate. It's just crazy. You can't really get it. You understand? So that's the thing. So I think these tests are available in the hospitals. I know not all hospitals do have. I'm not really sure of those things, but what I'm sure is that some of these tests are very cheap and easy to do. The oral test is easy. You administer, if the person gets allergic reaction, you note, you stop, you deliver your antihistamine, and you stop. You've identified. You understand? So it's quite cheap. And even the skin prick test is quite cheap. So I think maybe in another point, the health system needs to do better as well, which we always sing every day. We need to improve our health system. Like, it's very, very, very important. And I think that's the thing. Yeah, like if you see the hospitals in most of these developed countries, they have it's a key thing. It's part of your testing requirements. Like, you go to the hospital, they always ask you, okay, are you allergic to anything? Have you ever reacted to? Just give a hint. So they put them as probabilities. And so when they are running tests and the rest, they try to figure out from all your responses and see, okay, if they see there's a chance that there might be a link to this, then you run the test. That's how they do it here. So that's why you see they can really identify a lot of people that have allergic reactions and just help them through. So I think the health system also have to do better, as well as us informing and also spreading the word on allergens and educating people on food allergy.
Jasmine, you say you have a second question?
Yes, please. My second question goes thus. Out of the test, the 4 methods for allergens, that's the ELISA, PCR, LC, MS, LFG. I think was it the LFD or the MS that you mentioned? We can use something like a strip test where you
like a swab
drop at one edge and then it like a swap. Then from one edge it runs into the second stage and reacts with the I don't want to use the word chemical there, and if there's
you can use that. It's fine.
You then proceed and so on and so forth. Yeah. So you said that's the easiest one to use in factories, or can it be used in factory? It must be in the labs.
No,
and if yes, where are they accessible in Nigeria or over there?
Okay, yeah. Your first question, they can be used in your factories, like the LFD. Sometimes some people call them the swab test. So during cleaning, maybe what they do here, once they clean, they run a swab test, then run it through the LFD and see if these allergens are there. They can do it on equipment, they can do it in food. So they have testing the LFDs for food, and the rest they have the LFD for equipment or for cleaning. So they are available. They are let me not say they are cheap because there's money involved. So, but they are available in Nigeria. They are available in Nigeria. When I was in NAFDAC, I usually use the LFD for mycotoxin testing. Yeah, so, but because mycotoxin was like one of the key issue that was being tackled, so I used the LFD, the ELISA, and the MS for its analysis. So it's available in Nigeria. You get it. We have like the companies that produce this LFD, such like the Biofarm, the Neogen, they have representatives in Nigeria. Yeah, so you can easily get them.
Biofarm, Neogen.
Yeah, Biofarm. It's a German company. Yeah.
Bio.
I think I have a slide. Let me see.
Okay. All right. Thank you.
Yeah, let me see. I have a slide.
Okay.
Thank you.
Yeah, you're welcome.
Okay, while we are at it, a question was asked on the chat box. Okay. Sorry, for you to have an allergy, does it necessarily have to be consumed at all?
Very interesting question. It depends. Most of the time, the allergens are consumed. Yeah, they are consumed. But some other allergies, but we don't really consider them food allergies because they are pollens. They are environmental allergens that just induce itchy, rash, and the rest. But they are not really food allergens. But most allergens must be consumed. But there are some other types. There are some people, let me say, not other types, some people here that are kind of a bit more sensitive as compared to others. So they tend to get a bit reaction, yeah, on the skin. You understand? Like that's why the skin prick test works, because once you do that and you puncture the skin and put the allergen, because it comes in contact with the fluid. So if you have a perforation on your skin and there's a contact with the allergen, there's a possibility that an allergic reaction might be elicited. Yeah. So, but apart from that, most times it has to be consumed because it has to go into your system. It has to get in contact with a fluid, a bodily fluid, and the rest. So, but on the pure skin, it's difficult to induce allergic reaction, except there are pollens, which are environmental stuff that can just cause some allergies, but not food allergens.
Okay. I don't know if Immaculata is still here, if she still have a question, but why at that? I want to ask, is age a determinant of the development of an allergic distinct reaction? I'll explain further. My sister, while she was little, she wasn't allergic to milk, but now she's grown. She happens to be.
Mm-hmm. Yeah.
So is this something that develops over time?
Yeah, it's multi-directional. Let me see.
Okay.
It's age dependent sometimes. Yeah, because there are some people or some when you have young infants, like some of them infants are more susceptible to food allergy, so they tend to develop this allergy at very young age. Then some, it runs through all their lifespan till they get old and die. They are allergic to that particular food, while others outgrow allergy. So it all depends. So there's still a lot of studies in terms of this age-dependent allergy. Yeah. So, but it all depends. So some people don't get allergy when they're young, or develop it over time when they are old. So it all depends. So the only thing I can say, it's age dependent, because you can be allergic at a young age and outgrow the allergy, or you can be allergic at a young age and have it all through your lifetime, or you won't be allergic at a young age and suddenly develop an allergy over time. So it's definitely age dependent, and maybe there are some other factors that might be influencing that at different time of your life. So, but what we do, quite age dependent sometimes, but it's not specific. There's no well-defined reason for that.
Okay. Kikilomo, great presentation tonight. Kudos to our presenter and to Food Maestro team. Thank you so much.
Thank you.
And I don't know, is there any other question? All right. In the absence of that, we have a representative of NIFT to give us a closing remark. Or Jerry, do you have any other thing to?
Okay, nothing much. It's just thank you for being here, and it's my pleasure to discuss with you and share with you some information that I have, and so excited with the questions. Like these are some cool questions. Yeah, so I'm really happy to do this and always. And if you need maybe to get in contact, I can always share my information with Immaculate to share it to the group and as well. So just that.
We have a group, and you'll be added to the group. So I think that will be good. So if there is any question or anything, they can easily direct that to you.
Yeah.
And there will be a post test which will be sent to us, to our emails or the group during the week, at least to test our knowledge from what we have learned. And this time, 5 p.m. next week also, we go again for the good manufacturing practices training. So now we the representative of NIFT, I think Kikilomo, to come and give us the closing remark.
Yeah, thank you very much. Thank you very much. Immaculate, if I got it right. Am I
Yes,
right?
you do.
Yeah,
Yeah.
yeah. I'm honored to be doing this tonight. I'm standing in for my emeritus past president of the Nigerian Institute of Food Science and Technology, Mr. Wole Toye, who is unavoidably absent tonight. He was supposed to give this vote of thanks on behalf of our president, the president of NIST, the national president, Professor Abu. So I'm doing this on their behalf. I want to appreciate everyone for joining this call tonight. It's been a fantastic one. Mr. Jerry has done a
Yeah.
very beautiful job, and I think it deserves rounds of applause. Participants, if you don't mind, just send your emoji to appreciate Mr. Jerry. He has done fantastically well, and I was excited to know that you were in NAFDAC before you left Nigeria. I hope you're having a great time there. We are waiting for publication. Yes, publication on this.
Sure.
On
Sure.
this yeah, in the US. Thank you very much. And to the Food Master team, I must say thank you. This is the first time I'll be joining your training. I'm looking forward to the part 2 of this, and of course to other trainings that we'll be having. But I'm particularly excited about food safety. In fact, we just had a launching of the Association for Promotion of Food Safety and Improved Nutrition just this week, in Lagos, Nigeria. And, that association is open to everybody. We say food safety is everybody's business. So it's open to everyone, whether you're a nutritionist. So the issue of allergy, came up in that inauguration. The nutritionists and the dietitians that were available, raised, the issue of allergies. That not just food safety when it comes to hygiene and all of that. That nobody was talking about allergies. So I'm really particularly happy. This training is coming at a very good time. We're going to upscale this at chapter level. I'm going to upscale this, to other zones so that people can join trainings like this, and I'm sure that it will be a better world for all of us. So thank you once again, Mr. Jerry. You did
Thank you so much.
And a lecturer, sincerely. You on behalf of
Thank
NIFT,
you.
I want to say thank you. God bless you.
All right,
Thank you,
you're welcome.
Mr. Thank you very much. God bless you.
You're welcome.
Thank you. Thank you.
Yeah.
Thank you so much, Madam Kikilomo. I appreciate
Yeah.
this. All right. It's time to say good night or call it a day, whichever one. But have a wonderful evening. See you same time next week, okay?
All right.
Bye.
Yeah.
Bye.
Thank you very much. Bye.
All right.